In this episode of the Regenerative Health Podcast, I interview Professor Geoffrey Guy, medical doctor, former pharmaceutical executive, and billionaire philanthropic founder of the Guy Foundation, leading what I believe to be the world's most innovative research in health, that of quantum biology. In this interview, amongst a whole range of fascinating topics, we discuss the role of light in life, both endogenously generated light, also known as ultra weak biophoton, and exogenous light in the form of solar photons and their interactions. I believe the light and life story, explored through the lenses of quantum and circadian biology, hold the key to understanding and preventing chronic disease and optimizing human health. Hope you enjoy this podcast. Professor Guy, you've got a very interesting story that started with a medical school and detoured through the pharmaceutical industry and has now arrived at this fascinating work with quantum biology. So perhaps you could give us a brief idea of that story.
Well, I think the briefest idea is I often describe myself as a full-time medical student with a giant-sized chemistry set. But effectively, after a few years in medical practice, was training to be an obstetrician in London. I decided to take a sabbatical for a year to do some medical research in the pharmaceutical industry, and very fortunately with a pharmaceutical company in the south of France. It seemed extremely attractive at the time. I'd done a degree in pharmacology during my medical training, so that helped me a little bit. And I went off to France to work for a drug company called Pierre Fabre. I worked for Monsieur Pierre Fabre at the time. He had developed his business on the back of plant medicines, but was moving into new chemical entities. And my role was to take those first dose in man and then up into proof of concept. And I did that for a few years. Came back to the UK and worked for a company that developed control release morphine for terminal pain and other drug delivery products. And having worked for two private pharmaceutical companies at the tender age of about 29 or 30, I thought, well, I'd give it a go myself, and started my first pharmaceutical company, which was Ethical Holdings, which was a drug delivery business. We did that in the mid-80s. And in the 80s and 90s, drug delivery, control release tablets, different dosage forms, skin patches, those were very popular in terms of advancing therapeutics. And I came out of that company in about 97 and had previously considered which other products would benefit from modern drug delivery. And having worked with plant materials, opiates, for example, for pain, I'd focused on cannabis and cannabinoids in the early 90s, but had given that idea up when the regulators had told me that over their dead body would they allow me to do any development with a cannabis medicine. That had changed by the late 90s. The courts and the regulators were under pressure from a widespread use in cases like multiple sclerosis, the arthritides, rheumatoid and osteoarthritis, cancer pain, for example. And Her Majesty's government asked me whether I would address the issue of developing a medicine from cannabis. So I set up GW Pharmaceuticals, GW being my initials and those of the surname of my founding partner, Brian Whittle, so Guy and Whittle. And to cut a long story short, over 20 years, we developed Sativex for multiple sclerosis, and then the one that most people know about, which was Epidiolex, which is cannabidiol, for treatment of catastrophic epilepsies in children, now extended into adults. So we found ourselves really at the forefront, leaders in cannabinoid science, having turned it from being a cannabis plant being considered as an illegal plant to a medicine of the future. And I think it was either Scientific American, or Nature that described the cannabinoids as the aspirin of the 21st century. So we enjoyed very much the being able to research this complex area of medicine, very complex area indeed. But I was always concerned that being chairman of a NASDAQ company, by that time we were listed on NASDAQ, there was the chance that one day somebody would come along and want to take the company over. And I was concerned about really falling off an intellectual precipice. So my wife and I, in 2018, this was a number of years before we did sell the company, we established the Guy Foundation to look at the next generation of interesting research. And I often quote at the beginning of my lectures that Albert Einstein once said that if an idea wasn't at first considered absurd, then it had no chance. And I suppose I consider myself as the purveyor of absurd ideas the last 40 years. And so, going on from bringing opiates into modern use in the hospice and a terminal care environment, then making medicines out of cannabinoids, we thought the next step might be to look at something new and something over the horizon again. And that work really came out of some of the work that we were doing, trying to identify the mechanisms of action and the pharmacology of the cannabinoids.
Fantastic. So that is quite a pivot from cannabinoid and based medicines and pharmacology into quantum biology. So what was the, I guess, spark that made you look into perhaps from a biochemical to a more of a biophysical and biophysics perspective?
Having spent 40 years researching medicines and doing a large amount of pharmacology, when we were looking for the mode of action of the cannabinoids, it was difficult to pin it down in terms of classical Hillian pharmacology. And the more we looked at it, and the fact that the cannabinoids seemed to work across a wide range of therapeutic indications and work almost across all types of mechanisms and biological systems, we thought the link was always the bioenergetics, that every process, every mechanism of action requires energy. And we likened it to perhaps one arriving home, finding that one's remote control for the television doesn't work, and the transistor radio doesn't work, and your alarm clock doesn't work, and you work out you've actually bought a box of bad batteries. And so we began to focus on cellular bioenergetics. And I took the view, probably 15 years ago, that I thought the cannabinoids were working by modulating intracellular bioenergetics. And that would then be the balance of energy production and usage, energy dissipation, and of course, inflammation, that fine balance in intracellular energy and inflammatory milieu. So we began to think about the cannabinoids working at the level of mitochondria and at cellular bioenergetics. Initially, we were producing hypotheses that worked on standard or more classical thermodynamics. And at the same time, my colleague, long-standing colleague, Professor Alistair Nunn, had started to try and image cannabidiol inside mitochondria. And we were using 2-photon microscopy to do that, where one bombards the molecule with 1 photon, increases its excitation state a little, and then 20 femtoseconds later, you bombard with a second. That excites the molecule, and one could image it. And what we found is, as soon as we exposed the cannabidiol to any amounts of light and in certain wavelengths, it fluoresced. And what we realized, of course, is that we were dealing with quite a strong chromophore. And that very quickly made us really shift our thinking from classical thermodynamics into quantum science. And it was from that notion that we thought perhaps we ought to look more deeply into the underlying mechanisms of the fundamentals of how cells work and how all cells work. And that led us into quantum biology. And that was probably about 7 or 8 years ago, a couple of years before we started the foundation.
And fast forward to 2024, and you've had an absolute stellar lineup of cutting edge scientists present in your online series, including names like Dr. Nick Lane, Doug Wallace, Michael Schiffer. I mean, these are at the cutting edge of quantum biology. And so it seems to me, from a perspective also of trying to understand how to best optimize human biology and prevent chronic disease, that this is where we're at collectively, and this is nothing that is what is currently being addressed in I guess mainstream medicine or centralized medicine as it stands. But and perhaps we could start now with some real basic overviews of quantum biological concepts so that people really understand this at the base layer. So maybe how would you define quantum biology, the term, maybe to start with?
Well, I think in the simplest terms, biology is a study, and really uh, reverting to fundamental quantum mechanics and as it might impact on biological systems. I think that's the simplest approach to take.
Mm. And what are some of these quantum biological processes that are occurring inside cells?
Well, you could start in a number of places. I often start at the notion of the sort of the transition between the last 80 or 90 years of intense pharmacology, where medicine has been based almost entirely on chemicals, passage of chemicals, and chemical reactions, with some cross reference to electronics going on in nerves, for example. So I think the wave particle duality is the one that a lot of people have a difficulty in understanding. That perhaps the stick and ball structure of a molecule as we know it is only really the most highly probable structure when you transfer from a wave to a particle. And I think that once one can grapple with the notion of a molecule being more a wave, then this pinpoint target, this silver bullet approach that has been taken for the last 50 years or so in pharmacology, where the molecule has to arrive in a specific place with the appropriate stereochemistry, that disappears. Where, as I explained to people, it's more like filling a room with a gas as opposed to individual condensed particles. So
Mm.
that's probably where I tend to start when I talk to people. Then there's the issue of tunneling, that we can see that one waveform can tunnel through another, and that really brings us to the electron transport chain. And we, in the very early time with our foundation, I really set the objective of trying to visualize tunneling, and we haven't quite done that yet. And then there's the issues of superpositioning, and then the very alluring thought of entanglement. And in over the last 6 years or so, I think we've seen a lot of these issues that can now be placed in a biological setting, where prior to that, most people thought quantum mechanics had to be very cold and extremely dry. And now we know that quantum biology will occur in a warm, wet setting of the cells. And I think probably more recently was the notion of quantum spin. And that's what led us to thinking about the hazards of space health.
Do you mind explaining quantum entanglement and quantum spin a little bit more?
Well, as Einstein didn't like the idea of it. I think he called it spooky motion at a distance. This is the notion that you have two particles or waves, photons, electrons, protons, that are linked to one another in some way or other. And immediately one considers the notion of some information traveling between them. So if one has a electron spinning in one direction, and it has its entangled buddy elsewhere spinning in the opposite direction, if the first one changes, the second one is thought to change immediately, and without any perceptible change in its entropy or energy. And the notion that a lot of people have is that there's some sort of communication between the two. And I think that the best understanding is that these two entities, particles or waves, are mathematically correlated. They originated from the same point. They're mathematically correlated, and therefore will be mathematically correlated irrespective of the distance between them. And of course, a number of people say that one of them might be here, the other one might be the other side of the galaxy. And that tends to flummox most traditional conventional scientists straight away. But the recent experiments, as you know, have now pretty much demonstrated that entanglement occurs. I think most people are satisfied that entanglement does occur, but it does give the opportunities for us to think um, about how messaging and information could spread throughout biological systems far quicker with far less energy than had previously been thought. As you know, you and I, our brains are using about 20 watts of energy at the moment. A computer that could do the amount of computing that we do would probably need about 85,000 watts. So entanglement's very exciting. Also, it's interesting to understand how, for example, entangled photons might impact on molecules. And I don't know whether you know the work of Ted Goodson from University of Michigan. He bombarded a levoflurane anesthetic molecule with photons and established a conventional excited state. But when he then repeated that and bombarded them with entangled photons, he achieved a virtual excited state, which they considered would only exist about once in every 10 million years. So what intrigues us is when we're thinking about biophotons, those ultra weak emissions from mitochondria and from other parts of the cell, what intrigues us is to what extent any of those might be entangled and to what extent they might be altering the molecular interaction of incoming molecules. So that's something probably for the next decade to work out.
Incredibly interesting, and maybe it's a good opportunity to briefly describe the onion root experiment. And I know you had a prize named after that onion root, in terms of the history of this idea of biophoton research. And for the listeners who previously listened to my podcast, my episode with Dr. Jack Cruz, he mentioned Alexander Gurwitch specifically. So yeah, do you mind sharing your perspectives on that experiment?
Yes, well, when we started the foundation, we hadn't been particularly aware of Alexander Gurwich's work, but essentially in 1923, he was able to show that there was information transfer from the one onion root to another onion root, and to increase cellular division in the second onion root. And he identified this as effectively biophotons being released. And his view, they were actually in the ultraviolet range, interestingly enough. And he published that work in 23. There was a lot of skepticism about it. And it was really, I think it was only in about 1960s that I think his granddaughter finally produced the definitive work to show what had been done there. But the use of, or the notion that light will transmit information between cells or between organisms as a whole was, of course, used by Finson, for example, when he received the Nobel Prize in 1903 for treating lupus vulgaris with ultraviolet light as well. So in the early part of last century, there was quite a lot of interest in the use of light in terms of biology, in terms of therapeutics. But that, I think, got very much lost once we got into the post-war era of the pharmaceutical industries patenting molecules, and most of medicine research for the last 80 years or so has been based on purely molecular interactions.
And that's a point that I really want to emphasize, which is that this story appears to be one of both external light, which is the interactions of, say, solar photons on biological systems, but also one of internal light, which is the light being endogenously generated by structures like the mitochondria. And I believe Gurwitch, he used a piece of quartz to show that the biophoton emission was in the ultraviolet range, and that was actually blocked by glass. What's your perspective on this, I guess, distinction and the role of perhaps solar photons versus endogenously generated photons in the quantum biology story?
Well, biology is pleiotropic. It'll use every asset that's available, and time again. And some processes are conserved very highly throughout evolution from much lower order organisms. The sun and solar photons represent almost entirely the source of energy that we have used to evolve. And therefore, it seems to be no surprise whatsoever that there are very many systems in our body that are either using photons, communicating with photons, or indeed as part of their process, producing photons. Now, solar photons, of course, there's far more of them. On a sort of sunny day here in England, we've got about 10 to the 14 or 10 to the 15 photons per square centimeter per second in a normal daylight. I suspect in Australia it might be a bit more. And the biophotons, or the ultra weak emissions, we're down at 10 to 100 or so. So they are very, very weak indeed. So I think that the biophotons are really acting on structures very, very close themselves if they're acting physically, but of course there are the opportunities that they may well be entangled. The solar photons, of course, in different wavelengths can penetrate cells and whole organisms quite well, especially in the red range.
And specifically with regard to the frequencies or the wavelengths of light in these biophotons, as you mentioned, Gerwich's work suggested there was ultraviolet range. What is your perspective, or what have you noted in terms of what ranges of light are being emitted by different parts of the cell?
Well, different authors have produced different estimates there, and it seems to be across the range. And I suspect it might well be horses for courses. It may well depend on the purpose, function, and downstream objectives of a process. So we're still in the early phase of really trying to characterize these emissions. With our facilities now at Harwell, where we work with Central Laser Laboratories, we're able to detect down to very, very small number of biophotons. The equipment is in a basement, in a box, in another box, another box, to exclude all other extraneous light, and we have to settle the systems down. We have to be careful that we're not just detecting delayed luminescence that can confuse the situation. So we're wanting to characterize the biophotons, and the ideal outcome for us would be able to characterize biophotonic emissions from mitochondria that, for example, are distressed and are communicating mitochondria next to them. Now, we demonstrated that in a study that was published earlier in the year, Rees-Moll et al., where we showed non-chemical communication between mitochondria. So if that communication is with biophotons, it would be very, very interesting to understand if there is a specific characteristic and what information is being carried by a biophoton. And if we can characterize biophotons which are being released from mitochondria, perhaps saying, look, here we have a problem, saying to the next group of mitochondria, here's the solution, and by the way, here's a small packet of energy to deal with that problem, if that's what's going on, could we then, with our own lasers, emulate that biophotonic emission and re-impact or retarget mitochondria, so that we could produce something that we've called for the last 10 years mitotuning? Can we retune mitochondria by not only directing a range of photons at them, but photons that are specifically characteristic of those type of biophotons that would have been emitted by the mitochondria? So that's one of our objectives over the next few years.
Fascinating. And it gets to this idea of the function of the mitochondria. And I think at the really superficial level, what people are taught in university even and school is that these are just power plants of the cell, using that term. But looking at what's occurring on the electron transport chain, the kind of the exhaust of this mitochondrial engine obviously is water, and it's also these biophotons. I mean, they're actually being spewed out, so to speak, but perhaps not as waste. But what you're suggesting is actually perhaps as a signaling and information transfer mechanism.
Yes, look, at school we were all told that mitochondria were the powerhouse of the cell. And I think most life scientists would think of it as like a sort of a brazier, if you imagine on a picket line, everybody around warming their hands around a barrel with a fire in it, and just the energy is just being distributed in any direction. We initially began to learn it was really more like a sort of a mobile phone battery, which was discriminated as to how the amount of energy it would provide at certain times. Mitochondria seem to be able to discriminate which systems do need energy and which ones don't. And an example we give is if one has two neurons and requiring energy, if one gets energy and the other one doesn't, the mitochondria can actually alter behavior. And there has been work associating mitochondrial function with memory, for example. And we've moved even further on. And if you go to a power station, yes, there's a lot of energy generation, but probably the most important room in the entire power station is the ones where they have all the knobs and dials to decide how much power to produce, how much to dissipate, because they don't want to overfry the system, removing the rods from your nuclear power plant, just so that you're producing just the right amount of energy, and directing it to those parts of the grid that need it. And it's that information role of mitochondria that seems to come to the fore. And as you know, with plants, of course, they use the chloroplast to produce the energy, but they also have mitochondria. So it seems that mitochondria are communicating not only with other mitochondria within the cell, but of course cells exchange mitochondria exchange throughout the body, and there are actually free-flowing mitochondria in the plasma, although the jury's out as to whether those are fully functional or not. So part of the process of electrons tunneling their way down or flowing down the electron transport chain is that needs good quantum coherence. If there's any interruption or perturbation of that, then one has this sort of visualization of electrons spilling off the side, and that doing a couple of things, producing reactive oxygen species, and possibly a byproduct or a primary product of that is the release of photons. So every time you get ROS produced, you'll get biophotons produced. And it certainly seems to be the case that when mitochondria are distressed or reacting adaptively, if a lot of people describe mitochondrial dysfunction, what they really mean is it's mitochondria functioning perfectly well, but right at the extremes of their flexible limits. And when they're distressed, it seems that there's a much greater release of these ultra low emissions, these biophotons.
It prompts a fascinating question, and I had a funny image in my mind of The Simpsons and Homer Simpson at the nuclear power plant, with the donut and red lights going off. But where do you think in the mitochondria might that coordination decision be made in terms of perhaps the wavelength or the direction of light emission?
Oh, I think the answer to that is we don't know at the present. One can begin to sort of reverse engineer and speculate in so much as, can we identify pathophenotypes that may be associated with irregularities at different parts of the chain? And one of the culprits in the electron transport chain, I think, and if you listen to Nick Lane's talks on this, would be complex 1. So I think complex 1 seems to be, or the abnormalities or perturbations complex 1 seem to be responsible for a number of pathologies. And then, of course, down at the other end of the chain, the ATPase, of course, there are a number of mutations in the proton channels there that are also associated with illness. So we could sort of derive from circumstantial evidence which parts of the chain might be more important than other in communicating messages in terms of homeostasis and in terms of adaptive responses, by when they fail, where the adaptive responses fail to adapt appropriately, and then that appears as illness or disease. But I think we're not there yet, unless you have a better notion.
No, that question just came to my mind as you were speaking, and another question that really sparked my mind in terms of the endogenous production of light, and really when you mentioned the neurons, is this idea of endogenous light production as a signaling mechanism in the brain, and obviously clinicians in the audience will know that the brain exhibits a form of melanin called neuromelanin, which is
Yeah.
in Alzheimer's disease is sorry, Parkinson's disease is a pathologically lost. And the theory that Dr. Jack Cruz has proposed is that internally exhibited neuromelanin is acting as a form of way of capturing endogenously generated light in terms of signaling and other functions. And what and further to that point is that we have expression of these non-visual photoreceptor proteins like melanopsin throughout the brain. Do you have any insights or thoughts on those points particularly?
Well, I mean, clearly, our view is that light is important for the normal functioning of the brain, both endogenous and exogenous. To deal with the endogenous initially, some of the molecules to which you're referring could possibly better be thought of as antennae to really detect incoming photons and deal with them and respond appropriately. There's a lot more work that needs to be done to drill down and to silo down in a sort of an old-fashioned reductionist basis to work out what's going on immediately. But what we do know is that with exogenous light, there are a number of good studies now showing the change in neural function, neuropsychiatric function, with exposure to different wavelengths of light, mainly, of course, in the red, near infrared, and of course, much higher wavelengths. I always think it's strange that one of our most precious organs, the brain, isn't buried deeply in our body, surrounded by layers of fat to as a shock absorber, but it's sitting in exposed in with a very small thin piece of bone around it. And we now know that, of course, certain wavelengths of light, certainly the red light, will pass well into the brain, if not all the way through. And so to have an organ like the brain that is exposed to sunlight for the entire length of our evolution, and to know that also that the cells within, or the organelles within the cells within the brain, are also emitting light themselves. It seems to me that we're somewhat 2 or 3 decades late in thinking about this in the context of the advances made in medicine elsewhere.
Certainly, and that's the work of Scott Zimmerman, who's an optics engineer, showing that the sulca and the gyri are really optically optimized to concentrate near infrared photons into the gray matter. That really makes sense as well to me from a longer wavelength light point of view. But I'm curious if you have some thoughts on how shorter wavelength light, say ultraviolet, could be penetrating into the deeper inside the brain regions when we know that they're more superficially absorbed because of its wavelengths. Do you have any thoughts on that?
Yes, well, I think the shorter wavelengths scatter far more readily, and I know you spoke with Bob Fosbury, and he'll talk to you about first order scatter, and so blue and ultraviolet will not really penetrate very far at all into biological systems, whereas the reds and infrared will, and the invisible, non-visible light photons will pass all the way through. And it would seem, therefore, that over evolution, those systems that are going to use the energy of light and the information that can be carried by it and produced within light, will have effectively evolved around those wavelengths that can penetrate the whole of the organism. And equally, it's very helpful that although ultraviolet light was probably extremely important in the very early days of evolution to be the very high energy source. Of course, as soon as DNA became available to organisms, it was extremely damaging. And so one of the earlier processes in evolution must have been a way of dissipating energy not required. So if early organisms were utilizing and manipulating light energy and light information, there would have had to be a very good system to dissipate it, and so that organisms effectively didn't fry themselves. So I think medicine is beginning to understand a lot more about the therapeutic implications of anything above 600, 630 nanometers. And I think there's some catch up needs to be done in terms of the ultraviolet spectrum.
Yes. And the point about dissipation, I mean, I think that's a pretty critical again concept in quantum biology. Can you explain dissipation and this idea of a dissipative system?
Yeah, I mean, I think if you're going to tap into an endless source of energy emanating from the sun, one has to have a way of having a safety valve in terms of not being exposed to too much. Now, of course, with mitochondrial function, the mitochondria can uncouple so that you get futile cycling, usually resulting in increased heat in the cell. And it's often said that mitochondria are working at a much higher temperature than surrounding tissues. There was a paper a couple of years ago saying the temperature was nearly up to 50 degrees inside those. So I think any system that is using a freely available, abundant source of energy has to have a way of capping that energy and dissipating it appropriately. And obviously, in terms of oxidative phosphorylation, that would be done by uncoupling the energy production.
Mm. And a point to revisit quickly, the quantum coherence state. There's a suggestion that one of the roles of sleep and stillness at sleep is to facilitate quantum coherence. Do you any thoughts on that?
Oh, I think that's a very interesting notion. The brain, modern humans are, I think, subclinically inflamed. I often say that the average Roman soldier was about 7% fat or lipid, whereas the average human today is more. So we have a lot of ectopic lipid. That leads to increased intracellular inflammation. And I think it's fair to say that the humans as a whole, through their lifestyle, have mild mitochondrial dysfunction and intracellular inflammation. So we live in a far more inflammatory milieu than previously. The brain will also be the same, will be slightly inflamed, and it's only during sleep that process can be reversed, I think, and certain times during sleep, and that's very important to understand when that might be, and the work is only really its early phases. How sleep and quantum coherence go together, that's a very, very interesting thought, and something that I'd look into.
Mm. And so I guess understanding the mitochondria really, to me, is the logical focus, should be the logical focus of medicine, health, science as it stands. And I like to often reference Dr. Doug Wallace because his concept of this bioenergetic etiology of disease was one that posited that we shouldn't be looking at individual organ specific problems. We should recognize diseases of chronic nature in the kidney, in the heart, in the brain as simply organ specific manifestations of a bioenergetic failure. So how would you describe that key concept in terms of focusing on chronic disease and pathology?
Well, I think I agree entirely. When I was a medical student, and I think even now, one is really taught that the body is self-satisfying. It provides its own homeostatic environment, and that illness and disease is sort of the addition of something new, something extra has been added, and that's what's causing the problem. We take a slightly different view, and that is we're probably getting ill all the time. You've probably done that a few times since this interview started, but we have the processes to correct, to regenerate, and to repair, and we're doing that all the time. And that the emergence of some diseases, and a lot of diseases, is not really something new's turned up. Now we can think about, if one has been shot or there's a virus or something turns up, but generally it's the loss of that ability to continue to adapt and regenerate, which tends to get worse as one gets older. And all of those processes that we see in terms of mitophagy, in terms of apoptosis and regeneration of cells, and reusing some of the waste that's produced by after programmed cell death, those all roads lead back to the mitochondria, not only in terms of providing energy to do it, but the signaling and the messaging as to when to do it. I think that having a sort of a universal notion that mitochondrial dysfunction would lead to certain diseases in certain organs, I don't disagree with that. But we also know that mitochondria tend to be somewhat different. If you look at the mitochondria in the retina or in the heart or in the brain, their form and functioning can seem to be different to other parts of the body. So there is some organ or function specificity that would add to another layer if you've got a generalized mitochondrial dysfunction.
Mm. And that this concept of mitochondrial heteroplasmy, I mean, as I understand it, it's the accumulation of mitochondrial DNA mutations that are affecting essentially the translation of key proteins on the electron transport chain, and therefore the ability of the mitochondrion to produce energy. If there is and I guess the other point to make here is that if these are ancient bacterial remnants, which so we understand them to be, then their circular DNA is not protected by a nucleus, and the accumulation of reactive oxygen species from a leaky electron transport chain is therefore potentially rendering them more susceptible to damage and therefore accumulation of mutation. So can you speak to mitochondrial heteroplasmy and I guess the role it plays in potentially dysfunction or what you described as acting at the edge of its capabilities?
Yes, it's an area where very few researchers and clinicians really have a reasonable understanding. But it would seem that the human body adapting, especially if we've got a genetic disorder that is leading to a high level of mutation, it seems that we can tolerate really quite a high amount of mutation. Probably 50–60% of the mitochondrial DNA could be mutated with, but to maintain some function. But I think if we look more closely, we will see that more generally amongst the population, but it just doesn't represent itself as a pathophenotype. What we do see, of course, is a very close association now between mitochondrial DNA mutation and tumors and cancers. So when we look at cancers now, we can identify that some mitochondrial DNA mutations very, very regularly in those areas. So this is an area I think that is going to need a lot more research in terms of understanding how much background heteroplasmy there is, and at what point is there a threshold above which the mutated DNA determines the ultimate function in terms not only in energy production, but probably in information transfer. And it may be in a lot of conditions that the information transfer is the thing that suffers first. For example, in tumors, in cancer cells, where possibility is that energy production continues, but information transfer is suppressed, which allows then the tumor to grow somewhat rampantly without getting the feedback from surrounding systems and organisms.
And on that topic, I think it was Michael Schiffer's presentation where he described the charges of different cell types. And in tumorous cells, that they noticed a loss of negative charge. And
Yeah.
can you explain that concept or how you think about it?
It's fairly new to me that he was talking about both that in tumors, but also in wound healing as well, the differential charges. So we're beginning to understand a lot more. And this again comes away from this idea that everything is to do with the transfer of chemicals, the old view of a ligand sitting on a receptor, and that's about the only way that biology and medicine works. The charge across membranes in humans or in any organisms is enormous. The charge density across a membrane is something like 3 times that of lightning. So, Michael, I think is absolutely right in identifying that there is the differential charges, and if they change in, say, in tumors or in inflamed tissues, or where repair is occurring where there's been damage to tissues, then this is far more important than has been thought of before. And we have to somewhat get to grips with what's going on and how how can we either manipulate or enhance the appropriate changes in charge to accelerate or to improve healing and improve therapy.
Yeah, and that makes sense to me. And I think he also, as you mentioned, he described stem cells, and they have similar the lack of or loss of charge. So there's obviously a very intentional effect that the human body is using to when it comes to that. And the other like really big concept that I'd like to hear your thoughts on is biological semiconduction, and this idea that proteins that are in the body are essentially semiconductors. Can you speak to that idea?
Not greatly. It's not. It's not a very specific area of my knowledge at the present. We had a very good lecture by Judith Klinman a couple of years ago on that. What we are looking at, though, in terms of the interaction of protein with its environment and the notion of semiconduction, is looking at the layers of water, the interfacial water around proteins, and how that extends out way beyond the sort of normal distances that you would normally see bonding charges, for example. And our symposium series actually next spring will concentrate entirely on water order and the quantum characteristics of water. I think it's been completely overlooked in terms of medicine. We sort of think water's water and nothing's going on there, so let's think about the proteins. So I think when you look at the proteins, how they interact, we have to look at the interfacial water around the proteins and understand what's going on there as well. And of course, as you bombard that water with photons, you alter some of its characteristic, and including some of the it's thought also the viscosity of the water, which some people say has an impact on the speed or the readiness at which the ATPase nanomotor will rotate, for example. So we're going to be looking closely at water and certainly how it interacts with proteins. In terms of proteins and semiconductors, you'd probably have to talk to my scientists about that, I think.
Yeah, no worries. The water and light interactions are absolutely fascinating, and the idea that different light frequencies are changing the biophysical function of water. I mean, that is incredible, and the idea that even water is potentially has a information storage capacity, I think is one of those corollaries which is mind blowing, and again so far beyond the current state of the art in clinical practice and medicine. Maybe one more question specifically on the, I guess, the biophysics of this before we change tack. And can you speak to temperature and how temperature is affecting quantum processes and perhaps mitochondrial function?
Yes, it's a very interesting thought. For the last 20 years, I've asked all my colleagues a simple question. That is, how does a cell know what temperature it should be inside? Is it managed by a series of thermostats or whatever? Or is the temperature within a cell or within an organelle really the result of straightforward physics of a group of molecules in a vacuole being with a source of different sources of energy, both chemical energy and photonic energy, and electron energy? When you mix all of those factors together, does that determine what the temperature would be? What is the set point of the cell? Because if you think about conventional medicine and conventional chemistry, if you change the temperature, the equilibrium within all of the chemical reactions is going to change. And so, and we have this idea also that temperature is more important to quantum effects than heat. So I think it's really important to understand how temperature is regulated. Is it regulated, or is it just a result of the physics and chemistry that has been produced with the exogenous and endogenous energy supply? And as I mentioned early on, it's thought that the temperature within mitochondria is much higher than the surrounding materials. And we do know, for example, quite a number of plants, certain plants are exothermic, can produce temperatures regionally within themselves of 6 or 7 degrees higher than the surrounding or ambient temperature. So temperature, very important, but it's really understudied, and the understanding of how temperature is maintained or modulated within the cell is it really needs further study. And I'd come back to things like uncoupling. The result of uncoupling in the electron transport chain will be an increase in temperature, and that might be not a byproduct, but that might be the prime reason why we see uncoupling. If one alters temperature, then the quantum processes about which we're talking will themselves alter.
Yeah, and it makes me think of the clinical implications, which is people, when they get cold, quite often essentially stimulate thermogenesis in the body and the development of things like brown adipose tissue. And this has been used as an intervention to reverse metabolic syndrome and type 2 diabetes. And it's basically up regulating the thermal generating effect and helping pull things like that ectopic lipid out of the wrong place and essentially burning it. So I'm definitely intrigued at that temperature and how that is affecting things. And the other real interest that I have is the circadian system and circadian biology, and obviously it's critical to the whole organism function. Do you have any thoughts about how circadian biology and I guess the clock genes, which are nuclear in nature, in the nucleus, how are they interfacing with the mitochondria, and how are they affecting mitochondrial function and perhaps optimizing mitochondrial function?
An interesting thought. I mean, just going back to the point you made before, though, not all humans share the same cold genes. And therefore, for example, the Arabs, the Pima Indians, those that are suffering very high levels of metabolic syndrome, are unable to futile cycle. So when they get cold, they need to shiver. So it's interesting that it's not the same across all. In terms of, we're just, I think, at the beginning of understanding the more important role that mitochondria have in determining a circadian rhythm and the downstream effects that might have on aging. Again, most physicians of our generation would have been told that melatonin, for example, comes from the pineal gland, and that's the end of the story. We now know probably most of it comes from mitochondria. So mitochondria clearly very, very strongly involved in circadian, in the control of circadian rhythm. Now, how that interacts with these nuclear clocks, I'm not entirely sure, but I'm sure you have a lot more scientists with whom you speak, which will understand that better than me. But what we do see is the link now between the alteration in mitochondrial function controlling circadian rhythm and the red-blue balance to which we're being exposed at present. And I think you've spoken about this, and some of your guests have spoken about this, that there seems to be a sort of a red starvation and a sort of a shift towards blue, which is inappropriate for where the person was born or lives. And as we come under, that comes under stress, we might revert back to the red-blue balance that might have been needed nearer the equator a couple of 100,000 years ago, before humans were started to migrate. But that's the area that interests us in terms of the effects on mitochondrial function that have an effect on circadian rhythm, which have downstream implications for cellular and organism aging.
Yeah, thanks. It's such an interesting topic, and I really think that these type of questions are the ones that need to be funded and answered, especially in this day and age where we've electrified and lit up the whole of our nighttime in a very evolutionary and ancestrally inappropriate way. I wanted to ask you, what are some of your favorite or interesting insights from the series that you've done most recently with the amazing guests and researchers? Have you? I don't mean to ask you to pick from your favorite children, but which are some of the most enticing of these so-called crazy ideas that are alluring for you?
Well, I mean, one way of answering that question is you perhaps ought to read the new book that we've written, the quantum biology book. That sort of really covers those. What I see is a bringing together of vastly different disciplines in medicine. That's one of the things that you've mentioned. Our faculty has quantum physicists, mathematicians, astrophysicists, biologists, quantum biologists, and more conventional pharmacologists. And at one end, we're thinking about the work of Mike Levine and how it seems that it's not just genetics that are controlling shape and function. And to a certain extent, it may not be genetics at all in some instances. His work with planaria, where he can change the shape, the function of planaria without altering their genetics, where we can take a part of a planaria can be taught certain behaviors. You cut it in half, the head end grows a new tail, we all know that. The tail end grows a new head. But what happens is the tail end, when it grows new head, it exhibits the same behavior as it learnt before it was cut in half. So we think about memory as can be held somatically as opposed to just in the brain. So the notion of cells being far smarter in terms of memory and in terms of the stimulus for shape and production, possibly coming from the energetics surrounding them. And I talk a little bit about the quantum fractal. Does the quantum engine of life, in a way, produce a shape around which that organism grows? And each organism has a unique, each species has unique ATPase, for example. And we have a study running with Mike and Wayne Levine at the moment where we're looking to transfect ATP subunits and ATPase subunits and ATPase in a whole from one species to another to see whether we can transfer shape and function without altering genetics. So resetting a balance between the role of bioenergetics in producing form and function. And it was said that life is just an electron looking for a gradient. So to understand those gradients, how you drive those gradients. And then the thought that not only can these cells, small numbers of cells, remember processes which would lead to behavior, and that might be purely back to the point I made about allocating energy to different neurons or different neural processes to determine behavior. But of course, the work of Massachi only, the very early lecture we had looking at Physorum and the slime mold being able to effectively solve the traveling salesman conundrum. So the single cell, multinucleated single cell slime mold was actually able to solve a problem that has flummoxed most of the world's most powerful computers. And although it's not a quantum computer, it was behaving as a quantum computer. So now we see that cells not only have an ability to remember something and pass that on, but actually they can compute problems themselves. So computation may not only occur in the brain and through quantum wave theory, as you see in the brain, as John Joe McFadden will talk about, but that may also occur in other parts of the body. And you see that sort of in other animals, like things like squid and things like that, where quite a lot of processing can go on away from the brain. And then we think about talking about the work of Michael Cifra and the charge changes through around scar tissue and around damage and to tissues. You've probably seen the video of Mike Levine's anthrobots, which were just cells that have become like his xenobots. And that they, given without any prior knowledge, will run up and down a damaged tear in a neuron and over a 72-hour period repair it. And now, what attracted them was that just a sort of a normal physics-related response to a change in charge across a damaged membrane, and the natural response of the anthrobots was to do something with it. So we then think about what is the origin of the bioelectric templates, of which Mike talks about. What is the fundamental generator that can generate those bioelectric templates? And in generating the energy, does it generate a shapeform? And I talk in the new book a little bit about what I call a quantum fractal in terms of I explain it like, most people understand. If you put some iron filings on a piece of paper with a magnet underneath, the iron filings will take up the shape of the magnetic field. Well, in quantum terms, is there also a shape of these quantum fields, and is it around that biology forms? We take the view that life existed before biology. It's a self-replicating, non-dissipative, or self-replicating dissipative process, which decreased or didn't increase entropy as much as or as little as possible, and that biology just made that process portable. And this also leads to the notion that when we're looking for life outside the Earth, the problem is most people are looking for biology, but it may not be biology at all. It may just be very, very specific energetic entities that are able to self-replicate and carry information with them. And as you know, the physicists will say, to hold back entropy, you need energy and information, and that's why we think the mitochondria are so important in that role. So we then link that into where is the seat of this energy and information production, and all of those roads lead, in our mind, back to mitochondria. Although, of course, there's a very good notion now of non-mitochondrial ATP production, and so that's something else that we have to think about again. So ATP production may be more widespread than just purely in mitochondria, and then the next link is how we then link those bioenergetic processes to the myriad of pharmacological or biological systems that have been described over the last 60 years. That's what I'm trying to bring together, and from that come up with new, obviously, diagnostic modalities and new therapeutic modalities, which I think will occupy us for about the next couple of decades.
Yes, absolutely fascinating stuff. And I'd also point the listener to your 2016 review paper, The Quantum Mitochondrion and Optimal Health. And we touched on a couple of the topics in that paper, but I think there was still a lot more that we haven't. So I'm obviously mindful of your time, Professor Guy. So really appreciate you coming on and speaking with me. Where can people learn more about the book that you've written, and obviously more about the Guy Foundation and the series that you operate?
So the Guy Foundation, just Google the Guy Foundation and you'll land on our homepage. We have information there of our symposium series, the next one and the last ones. All of the symposia that we run two series per year, one in the spring, one in the autumn. I think there's about 55 videos now. They're about an hour each, with a range of scientists that form our faculty globally. Those are all on the website, but they're also on YouTube. There's a YouTube channel. And we also, on the site, describe the research that we funded both in the UK, US, and elsewhere. And we curate the research program to try and guide the process forwards. What we're trying to do with the foundation not only is funding research and leading it forward and bringing people together into a room, being in a virtual room, of course, that would never normally find themselves in the same room. And that's the important thing, is to try and overcome some of the really interesting thoughts. And we have quantum physicists and physicists, and on the whole, physicists do a lot of hypothesis generating, but occasional experiments. I think it took 40 years for the Higgs boson experiment to be undertaken. But once they've done the experiment, if the experimental findings correlate with the model, then it's given a tick and they move on. Life scientists are driven very much by experimentation. I think it was Boyle said that science is animated through experimentation, but you have to think about it a bit more. And of course, the grants and the way life science is funded on the back of experimental of proposals for experiments. But probably there's not enough hypothesis generation and thinking about it, life sciences. So initially we had all these different scientists, all with the same notion that quantum biology was of great interest, but the physicists saying, you haven't thought about it enough, and the life scientists saying, where's your data?
Yeah.
And now what we've hopefully managed to do is create those stepping stones in between to be able to have to bring these together. And that's, I think, is the fundamental role of it. The book, by the way, here's an advanced copy of the book, Quantum Biology. We wrote a book a couple of years ago about how we developed cannabidiol, CBD, which I think a lot of people around the world will know a lot about. But when I first started with our cannabinoid program, cannabidiol was described as an inert component of the cannabis plant. So we wrote a book about it, and there was a chapter in that book. That book was called A Worthwhile Medicine. And we wrote a chapter about quantum biology, and this was a few years ago. And the publisher said nobody understood it. Could you write a book that the layperson could understand? So we've written this book. It publishes in England, I think, next week on the 28th of November. And I actually saw it's on Amazon if people want it. So it's Quantum Biology by me.
Fascinating. Well, I'll definitely share that link out, and I'll definitely be grabbing myself a copy. So I agree that this is such a sorely needed area of education and research, and like you, I share the thought that this is how we help optimize human health. So, any final parting thoughts that you have for the listeners and maybe medical practitioners, doctors, medical students who might be listening?
Yes, I think the important thing is to level the playing pitch of knowledge and understanding. When we thought about the book, I've found over the last 6 or 7 years, it is much, much easier to explain quantum biology to a layperson who basically says, well, that's extremely interesting, or that's fascinating, and this happens and this happens. Whereas when we start with conventional traditional scientists. I can sometimes not get past the first sentence before the scientist says, "Oh, whoa, hold on, that's not what I learned." So what I would say is level the playing pitch. Just sort of set to one side the more conventional thoughts about biology, about physiology, about pharmacology, and understand a little bit of what really absolutely drives us. And I often say to people, imagine you and I had been having this conversation, and say, for example, this cup, for example, had hovered there for the whole conversation. Okay. Someone might say to you, well, what did Dr. Guy, what did Professor Guy say to you? And you say, I have no idea. I couldn't take my eye off this cup. It was suspended there. I had no idea how it was suspended there. It absolutely was miraculous. And I say to physicians, I say, when you look at life, when you look at humans, it's as confusing, as complex, and as miraculous as that cup being suspended there. And it's worthwhile spending a bit of time, if you're a physician or a life scientist, not just to dig straight into what's a disease and what's an illness and how we treat it, because medicine is mainly pattern matching. It's a lot of pattern matching. But actually go back to fundamentals and say, well, if I'm treating these organisms, these humans, it might be worthwhile understanding what drives them, what actually makes a cell work. And what we now know is it doesn't stop just at chemicals. It doesn't stop just at nerves traveling rather slowly up signals traveling rather slowly along nerves. That quantum biology or quantum science and quantum mechanics has a fundamental role in the fundamental workings of us allowing electrons to flow down a gradient. That's what life is all about. And I say to these doctors, everything from the chin downwards is orientated to finding glucose for the brain, because the brain needs glucose, we need oxygen, and those two are incorporated in maintaining these gradients. And so that's what I'd say to doctors, is sort of just open your mind a little bit and think about and read this quantum science, and try and escape the norms of medical teachings of the last 50 years or so, which has been extremely syllogistic. There's a lot of syllogism. There's a lot of if this, then that. And a lot of medical thinking relies on the modern day doctor considering that they understand the mechanism of action. And frankly, if they don't understand the mechanism of action, they can't get involved with therapy. And I tend to be a phenomenologist. I observe phenomena. I believe the patient, why doesn't somebody else? And begin to manipulate and use that phenomena and see if we can alter it, see if we can emulate it and harness it for therapeutic or diagnostic purposes or prophylactic purposes. And so I think one has to set aside that syllogism of intellect and just be rather more accepting that these rather odd things, very weird things that go on in quantum physics, just accept that, okay, that's fine. Let's accept that happens. Now, what is the impact of this in human physiology and human biology? And to what extent can we understand it, grapple with it, and manipulate it? And that's obviously something that we've published recently in our space report.
Yes, and we didn't get into the space report. We maybe that's a topic for another episode, and I'll point people to my previous episode. I did a presentation about the space report, but really, I couldn't agree with you anymore. And I really think what you're doing and the emerging field of quantum biology is the core of what is a decentralized movement in science and health, and that decentralization is answering questions that centrally funded, centrally administered, maybe even gated institutions haven't given us answers to these complex questions. So, very much appreciate your work and the work of the foundation. I'll be following it very closely into the future. So, thank you very much, Professor Guy, for your time and for speaking with us.
Thank you very much. It's been fascinating and intriguing. Thank you.